I'm Dr. Scott Greilich, a senior scientist at Immunis. Today I will be discussing our study published in Aging Cell titled Stem Cell Secretome Treatment Improves Whole Body Metabolism, Reduces Adiposity, and Promotes Skeletal Muscle Function in Aged Mice. These data represent an extension of the results from our geroscience publications, where we examine the effects of secretome on muscle mass and function in aged mouse models of disuse and recovery. We have a separate video on our channel discussing our publication in geroscience. As we age, we progressively lose muscle mass and strength, known as sarcopenia. In addition to this muscle loss, our bodies also begin to deposit increased fat tissue throughout our bodies. Our overall physical function and independence as we get older is heavily dependent on our muscle strength and metabolic activity, making it imperative for researchers to find therapeutic treatments that can reverse age-related muscle decline and enhance metabolism. Standard changes to diet and exercise are the main approaches to increase muscle mass and combat obesity, but these options may not be feasible for the elderly. There are also no drugs available to enhance muscle growth or manage metabolic dysfunction in adipose tissue, also known as fat. Therefore, there is a great interest in creating a treatment that addresses sarcopenic obesity to improve physical activity and promote quality of life. Immunis is attempting to address these concerns by using human cell-derived secretome to reverse age-related muscle loss and metabolic dysfunction. The secretome comprises of all factors released from a cell. It includes secreted proteins, lipids, growth factors, exosomes, and many other signals necessary for cell-to-cell communication. The nature composition of a secretome is highly complex and dependent on the environmental conditions and cell type from which it arises. Immunis' research team developed a clinical-grade human cell-derived secretome from our partially differentiated stem cells. Our secretome is enriched with extracellular vesicles and growth factors previously published to promote cytoskeletal remodeling. Immunis collaborated with Professor Micah Drummond of the University of Utah to study how Immunis' investigational secretome alters muscle and fat content in aged mice as a potential treatment for obesity and age-related muscle loss in humans. Let's dive in. Male mice, aged 22 to 24 months, which is equivalent to a human in their 60s, were assigned to either secretome treatment or saline control groups. The mice received injections of secretome or saline in their quadricep muscle twice a week for 4 weeks, totaling 8 injections. To conduct whole body metabolic measurements, the mice were housed in a comprehensive lab animal monitoring system, or CLAMS for short. The CLAMS were used to calculate the mouse's respiratory exchange ratio, or RER, by measuring the ratio of CO2 production to O2 consumption. RER indirectly shows the muscle's capacity to acquire energy. At high RER indicates that carbohydrates are being predominantly used for energy, whereas a low RER suggests lipids are predominantly used. Energy expenditure was calculated by dividing total heat production within the CLAMS by the mouse's body weight. Mobility and physical activity were determined by adding the number of times the mice intercepted light beams from the X, Y, and Z axes. Food intake was also measured by the change in mass of the food consumed. Following the 4-week treatment period, secretome-treated mice had significantly greater oxygen consumption and increased carbon dioxide production within a 24-hour analysis window. The RER was also greater for the mice who had secretome treatment. Additionally, there was an increase in energy expenditure, physical activity, and food intake for secretome-treated mice versus controls. Does the secretome affect the metabolism of aged mice? Yes. We have shown that secretome-treated mice consume more oxygen, release more carbon dioxide, expend more energy, move around more frequently, and consume more food, which is likely fueling their activity. In conclusion, the secretome helped the elderly mice become more physically active. Next, the researchers conducted tests to thoroughly capture any physiological changes with secretome treatment by examining their whole body tissue content. Secretome-treated mice showed a progressive increase in body mass, but an overall decrease in the percentage of body fat. These data are supported by the increased lean mass and reduced fat mass with secretome treatment versus controls. To assess the mice for physical fitness, they next conducted a series of strength, balance, and coordination tests. At week 3 and 4, grip strength increased from baseline levels for secretome-treated mice only. For the balance test, secretome-treated mice outperformed control mice as determined by the change in rotor rod performance times from baseline. Lastly, at 4 weeks, the secretome significantly increased the mass of both quadricep muscles compared to controls. Fat mass located near the hind limbs was also reduced in secretome-treated mice. Does the secretome induce physiological changes to aged mouse muscle? Yes. Our data shows that secretome treatment increases lean body mass, reduces fat mass, decreases percentage of body fat, enhances grip strength, improves balance, and increases muscle mass. Together, these data demonstrate that the secretome induces positive physiological change to fat and muscle tissue in aged mice. Next, the researchers investigated the effects of secretome treatment on quadricep muscle morphology and cell tissue remodeling. Secretome-treated mice had greater average muscle fiber area and diameter compared to controls. The secretome treatment did not result in any change to the percentage of type 2A and type 2B muscle fibers. However, secretome-treated quadriceps showed a rightward shift in size distribution for type 2B muscle fibers compared to controls. Specifically, the secretome treatment group had a significantly greater proportion of type 2B muscle fibers that were larger than 2,500 micrometers squared. Does the secretome affect muscle morphology? Yes. In aged mice, secretome treatment increases total muscle fiber area and increases the proportion of larger sized individual muscle fibers. To summarize, these data show that the secretome promotes muscle fiber growth by enlarging muscle fibers. In addition to observing morphological changes in aged mouse muscle treated with secretome, we also observed that secretome induces cellular remodeling. Compared to controls, secretome treatment increased the number of PAX7 positive satellite cells within the quadricep. Satellite cells are muscle stem cells that are responsible for muscle regeneration and growth. Secretome-treated mice also have more CD31 positive cells in their muscle tissue, which marks capillaries, suggesting secretome treatment may also support greater vascularity in the muscle. Lastly, the researchers calculated collagen turnover by determining the ratio of collagen hybridizing peptide, indicating collagen breakdown, compared to collagen 4, indicating collagen synthesis. Secretome-treated mice had a greater BCHP to collagen 4 ratio, indicating greater collagen turnover, which is an essential part of muscle regeneration. Does the secretome induce cellular remodeling? Yes. Our data illustrates that the secretome treatment increases the number of muscle stem cells, increases capillarization, and increases collagen turnover. The outcome of these experiments indicates that the secretome benefits the muscle tissue environment in favor of muscle regeneration. As our initial analysis showed, the secretome has robust effects on whole body fat content. The team investigated whether these effects were on fat in specific tissue compartments. With secretome treatment, there was a greater proportion of smaller sized fat cells within the muscle compared to controls. These data were supported by the increased expression of hormone-sensitive lipase, or HSL, which promotes lipid breakdown, and the decrease in total triglycerides and ceramides within the muscle. High levels of triglycerides and ceramides are common in those with impaired metabolic health. Lastly, there was no change in lipid content or fibrosis within the liver, which is the organ responsible for breaking down fat. Does the secretome alter the fat content within specific tissues in aged mice? Yes. We have shown that the secretome-treated mice have smaller sized fat cells within the muscle tissue, increased expression of HSL protein to break down fat, decreased muscle fats such as triglycerides and ceramides, no change in fat content or fibrosis within the liver. In brief, secretome treatment reduces fat in specific organs and tissues within the body that are commonly associated with metabolic disease. The team next used 2 immortalized mouse cell lines to investigate direct and indirect effects of secretome treatment on muscle and fat cells in vitro. For direct secretome treatment, the cell lines were incubated in control media or media containing secretome for 24 hours. For indirect secretome treatment, cells were treated with control media or media containing secretome for 24 hours, followed by an additional 3-hour incubation in fresh cultured media. These samples are referred to as control CM or secretome CM. The culture media is then harvested and used for a separate 24-hour incubation on the cells in vitro. This 3-hour incubation strips the cells of secretome and helps determine whether the effects on muscle and fat are a direct result of secretome factors or an indirect effect of the molecules subsequently secreted by the cells. The researchers determined that direct secretome treatment increased muscle area and fusion compared to controls, whereas indirect secretome treatment only increased muscle area compared to its respective control. Secretome treatment also directly and indirectly reduced lipid droplet area of fat cells compared to controls. To determine a possible mediator of the indirect benefits of secretome, the team then measured expression of IL-6, a cytokine released by skeletal muscle during exercise. Secretome culture media showed more than a 3-fold increase in IL-6 expression compared to control cultured media. Does the secretome affect muscle and adipocytes directly or indirectly? Both. Our in vitro studies reveal that secretome treatment directly increases muscle cell area, directly increases fusion of muscle fibers, directly reduces lipid droplet size, may mediate effects through the increased IL-6 expression. Together, these findings suggest a combination of direct and indirect effects of secretome on muscle and fat tissue. Although the exact mechanism of driving muscle growth and fat shrinkage warrants further investigation, secretome treatment is at the root of these positive outcomes. In conclusion, maintenance of muscle mass and healthy fat content positively influences our health span. We showed that in preclinical models, the secretome increases muscle size and strength, promotes extracellular matrix remodeling, and decreases fat. These data provide a promising basis for therapeutic developments targeting metabolic dysfunction and sarcopenia in at-risk aging populations. We look forward to pursuing these investigations further and gaining a better understanding of how the secretome can support these important elements of health.